بسم الله الرحمن الرحيم به نستعين اللهم علمنا عنك يا رب فهمنا عنك يا رب. Day 9 of my journey to learn bioinformatics using ChatGPT. It is titled Introduction to SNP, Single Nucleotide Polymorphism. What is single nucleotide polymorphism? It is a variation in a single nucleotide like A, T, C or G at a specific position of the genome. Because I said it is a genome, so these variations can be present in the coding and non-coding part of my genetic. These SNPs are the most common type of genetic variations among individuals. Okay. What are the key points to learn? Number one, what are SNPs? SNPs occur when a single nucleotide in the genome differ between individuals. Okay, so here have example. We said genome. This means that this SNP can be present in the coding or the non-coding part of the genome. Okay, this person number one. These are the same genetic area. Okay, let's say it is gene of insulin. For an example, okay, we found this variation in person 1. This is the sequence. In person 2, this is the sequence. When we compare the two sequences, we found that this C nucleotide is replaced in this person number 2 with T. Okay, so he say this SNP, so it is an only nucleotide that is replaced with another. So C is replaced with a T, making what is called an SNP. Okay, where do SNP occur? It can be in coding regions or in the non-coding regions. In coding regions, these are the called exons. Exons, they are the area of the genome that will be expressed, will be transcribed into messenger RNA, then will be translated into protein. Okay, this is the definitions of a coding regions. Okay, so if a SNP is present in these coding regions, it can affect the protein. It can affect the protein. Okay, so it change a sequence of the sequence of nucleotide will lead to change in the amino acid will be put in this polypeptide chain, which may affect the structure of the protein. Building on changing the structure of protein, it can change its function. Okay, it also SNPs can be in the non-coding regions. Non-coding regions called introns. These introns are not fully understood the function, but right now we are studying it excessively and found its regulation. It contains a regulation region. So any change, this SNP, when present in these non-coding regions, it may influence, it may affect the gene expression of the genes around this region. Okay, so what we understand about it? Number one, intron, it are not fully understood its function. Until now, we know we reach that it is containing a regulatory regions that affect the expression of the surrounding coding regions. So if a SNP present, it influence what? The gene expression. What is meant by gene expression? Okay, we have a genetic material called gene. These genes want to express this information that is represented in the sequence of nucleotide. This expression form a functional product. This functional product can be protein or can be RNA. Examples of a functional RNA are ribosomal RNA, tRNA, microRNA, long RNA. These these RNA are not translated and as such have a function in our body. Okay. Another title, why are SNP are important? Why we care to study it? Number one, disease association. We found that specific SNP in a specific site are linked to disease like cancer. So, a specific type of cancer, we link it is always associated with presence of a specific SNP in a specific site. So, we can help us in expect that this person, if have this SNP in this specific site, it will in the future predispose to have this type of cancer. Okay. SNP also is associated with other diseases like diabetes, heart disease, and many other diseases. Number two, pharmacogenomics. What is pharmacogenomic? It is the presence of SNPs that affect how individual responds to drug. This idea, many persons have the drug, okay, have this drug for treatment to these conditions, but not all the person have the same response. And we ask why. Why some having exaggerated response while others are resistant? Okay. So one of explanations, one of the area that we research in is pharmacogenomic. It is this person having this genetic predisposition to exaggerate its response or resistant to this drug. Okay, so let's study what are these genomics that can affect our response to drug. One of them is SNPs. So from the start, you can detect SNPs that give you a future picture about the response of this person to this drug. What else? Population genetic. It helps in trace ancestry and evolution history. How this? There is a specific step that is widespread in a specific population, in specific races, in a specific family. So we can trace these SNPs and understand the ancestry of each person. Okay, what are the tools that is present to study SNPs? Number one, databases. There is a database for single nucleotide polymorphism. I take the name of this single nucleotide polymorphism. Okay. And this is the database that having a national library. I will do exactly what I have done. I take this, its name, I take a copy. Okay, I go to my browser. Okay, so it is the first one. Voila, this is the database. I'm so sorry. What other tools to study the SNPs is genome-wide associated study. They are studies that link the SNP to disease. Okay, I do the same. I take it copy. I Google it, and this is the National Human Genome Research Institute Genome-Wide Association Study. Okay. What else? So these are the tools. What else? Application. In what we use SNPs? Number one, personalized medicine. It is developing treatment tailored to individual. What we are doing now, it is one treatment or one size fit all. Okay? No. The next step in future is tailoring the medicine to the individual about its using its genetic profile to provide a proper treatment for him. Through using what? These genetic variation that predispose or affect its response to treatment. Okay, like pharmacogenomics. Okay, what else? Disease diagnosis. Identifying genetic predisposition to disease. As we said, we link some single nucleotide polymorphism in a specific site with specific type of diseases like cancer. So if I scan this person for these SNPs, I will have a report about the diseases that this person is predisposed to. So I can form a plan to keep an eye on him, use a prophylactic drugs, use a more visit to the clinic to detect these diseases early. Okay, what else? Evolution study. We said that some SNPs are present in some families, in specific races, in specific populations, so we can use it in understanding the genetic diversity across population. Okay, the last one is the task. This task is number one, explore this single nucleotide polymorphism SNP databases. We will visit the site and search for a SNP called this. Okay, I will copy this, I copy this SNP, I'm so sorry. I will go to the database of single. I will copy in the search. Okay. I will go for search. Okay. And another part is the view information related to this SNP. It is present in Homo sapien. Okay, what are the variant type, what are allele, chromosome, gene, functional consequence, okay, clinical significance. So this is the first application. The other application is optional, which is optional. Read about how SNP are used in personalized medicine or ancestry testing. Okay, I am interested in personalized medicine. Ancestry testing is not my interest, so I love to read more about how SNPs used in personalized medicine. I will take this question, copy. Okay, I will go to another AI tool called Perplexity. Okay, I will give it the question. Okay, and search. I used the deep search, so it takes a lot of time. You can use whatever you interested in. Okay, so I have a report about the role of single nucleotide polymorphism in advancing personalized medicine. I can read what I am interested in. سبحانك اللهم وبحمدك أشهد أن لا إله إلا أنت أستغفرك وأتوب إليك.